So now we are going to study in detail about the neurulation process. Now this is the developing embryo. This is entire developing embryo, and the surface is the area is called as a neural area. You can see the outward projection is the neural groove. I will see this is the vertical view. This is the vertical view. We can see the horizontal view now. Now the outer blue one is called as an ectoderm, which is modified as a neural ectoderm. Then this is the mesoderm. You can also see here, this is the ectoderm. Then this is the mesoderm. Here the paraxial mesoderm, this one, and this is paraxial mesoderm, and this is the intermediate mesoderm. Okay. Now about the mesoderm, it is an ectoderm. So in the ectoderm, it will modify as an neuroectoderm, and it will form a inward projection, that is a groove-like structure. Here groove-like structure, inward projection. Okay. Now you can see this is the neural fold, neural groove, and this is the primitive streak and the primitive node of the embryo. This is an embryo. Now the outermost layer, this is an ectoderm. The ectoderm is modified as a surface ectoderm and as a neural ectoderm. From these, it will show the inward projection like this, inward projection, and the inward projection is called as a neural groove. And on the anterior sides of the neural projection, on both the sides, they are called as a neural fold, and the cells in between these is called as a neural crest. Okay, neural crest cells. Now here it is a notochord. Now you can see this is a surface ectoderm, the ectodermal modification. Now the neural crest cells are moving out from both the sides of the neural groove to the process called delamination, that is the removing of cells from the main layer. The cells will be removed from the main layer, individual cell movement. Okay. Now this is how it looks. You can see the neural groove, deeper invagination, and here one mechanism, that is same type of cells will interact with the same type of cells. Okay. So after differentiation, what happens? The cells, the neural crest cells, they are differentiated one type of cells. The neural fold cells, one type of cells, and the neural groove cells, one type of cells. So what happens? Whatever the neural groove cells are there, they will fuse like this. Neural fold cells fuse like this, and the neural crest cells like this. It will lead to the formation of the entire round-like structure. This is circular structure. You can see this neural fold approaching on each other. So this is, they will come closer in contact. This neural folds, they come closer in the contact by forming a round structure, that is circular structure like this, and it is now called as a neural tube. Okay, it is now called as a neural tube. Now you can see here the entire, this is the neural tube, the entire structure. The in between is called as a neural canal, and whatever the cells which are moved with the process called delamination, and they are known as neural crest cells, and this is the neural ectoderm. Neural ectoderm. So now you can see this is modified as an epidermis. Now this is the neural tube, and this is the notochord. This so every cells in the developing process have a specific importance. Okay, the neural tube cells have different function. The crest cells have different function. The surface ectoderm cells have different function. So everything has a different function. So here neurulation is of two types. That is primary neurulation, that lead to the formation of the brain and spinal cord. Spinal cord, that is nothing but the central nervous system, and second process is secondary neurulation, that lead to the formation of the sacral and the coccygeal cord. Okay, so now we'll see what are the derivatives of the neural tube structure derivatives. Just now I told you, central nervous system, it is forming the brain and the spinal cord, central nervous system. Then it will lead to the formation of retina, that is eye. So both the central nervous system and the retina, they are derivative of ectoderm. They are originated from the ectoderm. They are ectodermal in the origin. Now we will see about the derivatives of neural crest cells. So neural crest cells, they are forming the spinal ganglia, that is dorsal root ganglia, then ganglia of autonomic nervous system, then ganglia of cranial nervous system, that is cranial nerves, 5, 7, 9, 10, then peripheral, then they are forming the peripheral nerves, then coverings of the brain. What are the coverings of the brain? There are 3 coverings of the brain now. Meninges, the coverings of the brain, the spinal cord, the pigment cells which are present in the brain, and the suprarenal medulla.